Welcome to the pod Biomanufacturing and Fermentation Technology. In this episode, we translate microbial science, process engineering, and manufacturing realities into insights that scale.
If you look in your medicine cabinet right now, think about the last time you got a shot at a clinic, you probably picture a glass vial.
Right, just a tiny sealed bottle filled with clear liquid.
Exactly. And for decades, that physical vial has kind of represented the absolute cutting edge of biological manufacturing.
Yeah, it really has.
But what if I told you that to understand where 21st century medicine is actually heading, you need to completely forget about the glass vial.
Oh, totally. You have to throw that image out.
You really do. You need to stop thinking about liquids and, start thinking about computer code.
It represents a massive paradigm shift. I mean, we are moving away from treating biology as this physical substance that we just, farm and harvest.
Right.
And we're moving toward treating biology as an information layer, something we can actively program.
And that is exactly our mission for today's deep dive. We are exploring a truly fascinating document. It's simply titled mRNA Vaccines-V2.difo-X.
And it is a dense read, but so worth it.
Oh, absolutely. Let me tell you, the insights in this source completely upend how we should think about the future of healthcare.
They really challenge all the old assumptions.
So we're going to unpack how medical science has successfully deployed a programmable information layer into the human body and, at an industrial scale.
Which is staggering when you really break it down.
It is. We'll look at the sheer engineering physics of how it's done and what this radical shift means for your own future. Okay, let's unpack this, because the core breakthrough here is something I want to try and explain with an analogy.
Oh, I am always ready for a good analogy to ground these concepts. Lay it on me.
So think about buying furniture. Traditional vaccines, where we use inactivated viruses or grow viral proteins in these massive steel vats, they're kind of like shipping fully assembled furniture.
Like buying a couch off the showroom floor.
Exactly. You build the whole couch in a factory, you load it onto a truck, and you ship it to the body.
Right, and then the body's immune system sees the couch, recognizes its shape, and learns how to defend against it later.
Spot on. But mRNA is an entirely different logistics model. mRNA is like shipping the IKEA instruction manual directly to your cells.
That's a great way to put it.
Yeah, you aren't sending the couch at all, right? You're sending the blueprint.
Oh.
And you're letting your own cells use their internal workshops to build the couch themselves.
To build on that analogy, shipping the instructions sounds brilliant on paper. And the source document actually points out that this blueprint concept has been around since the 1990s.
Wait, really? The 90s?
Yeah, but, conceptually simple does not mean practically easy. Taking that from a whiteboard theory to a global medical reality required scientists to solve 3 incredibly difficult, interlocked engineering hurdles.
These were not small hurdles.
Not at all. First, they had to stop the body's innate immune system from freaking out and just shredding the mRNA blueprint the second it arrived.
Right, because the body hates foreign stuff.
Exactly. Second, they had to sneak that fragile mRNA inside the cell's actual workshop, which is called the cytosol.
Getting past the bouncers, basically.
Yeah. And third, they had to figure out how to manufacture this delicate code at a massive scale with flawless consistency.
Okay, let's focus on that first hurdle for a second. If you think about human evolution, the body is essentially a fortress, right? It's designed to attack and destroy foreign genetic material.
That's its main job.
I mean, a virus is basically just rogue genetic code trying to hack into our cells. So here's my pushback. How on earth do you slip an artificial genetic blueprint past a security system that has been evolving for millions of years to destroy exactly that?
What's fascinating here is that the scientists didn't try to overpower the immune system with brute force. They cleverly disguised the code.
A disguise. How does that work?
Well, our cells have these internal sensors. They act like molecular tripwires that are highly tuned to detect foreign, unmodified RNA.
Like a motion detector for viruses.
Exactly. So to bypass those tripwires, researchers made very specific chemical modifications to the mRNA itself. The source highlights a crucial breakthrough involving something called a nucleoside modification.
Right, I remember reading that part.
Specifically, it's called N1-methylpseudouridine.
Yeah, the text gets really deep into the chemistry there. For those of us who aren't molecular biologists, what does swapping out a nucleoside actually do?
Okay, think of the genetic sequence as a sentence, and the nucleosides are the letters making up the words.
Okay, following you.
They found a way to slightly alter the physical shape of one of those basic chemical letters. Imagine swapping out a standard letter U with the exact same letter, just printed in a slightly different font.
Oh, that's clever.
Right. The cell's machinery can still easily read the word and understand the instructions, but the immune system's security scanners don't recognize that specific font as a threat. It slips right by.
Molecular camouflage through typography. I love that.
It's beautifully elegant.
But the document also mentions they optimized other parts of the strand, like the 5' caps and untranslated regions. If the nucleoside is the font, what are these other modifications doing?
They provide the packaging and handling instructions. The 5' cap is basically a chemical stamp placed at the very beginning of the mRNA strand.
Like a barcode.
Kind of like a priority mail sticker. When the cell sees that cap, it knows this strand is important and shouldn't be degraded.
Oh, okay. And the untranslated regions, the UTRs.
Right, the UTRs sit before and after the actual protein building code. They are essentially instructions for the cell's machinery on how fast to read the blueprint and, how many times to build the protein before throwing the instructions away.
So by tweaking all of these modular elements, the caps, the UTRs, the letters themselves, they shifted the mRNA from being perceived as a glaring danger signal to a perfectly normal, highly efficient request for the cell's factory.
Exactly. It just looks like everyday business to the cell.
But that brings up the physical reality of the body. Even with the perfect camouflage, this blueprint is notoriously fragile.
Incredibly fragile.
Right. If you just injected raw mRNA into your arm, the enzymes in your bloodstream would chew it into pieces in seconds. So how does this fragile string of code physically survive the journey to the cell's workshop?
That requires an engineering marvel in its own right. It moves us from the blueprint to the delivery vehicle. The chariot for this code is the lipid nanoparticle, or LNP.
The famous LNP.
Yeah, it is basically a microscopic bubble made of fat, but it's a highly engineered multi-component system. The source breaks down a very specific mixture of these.
It lists ionizable cationic lipids, helper lipids, and pegylated lipids.
Yeah. Yep, that's the recipe.
That is a lot of lipid terminology. Let's break those down, starting with the ionizable cationic lipids, because the text seems to suggest they are the real magic trick here.
They really are the driving force of the entire delivery system. These specific lipids have a unique property. Depending on the acidity of their surrounding environment, they can change their electrical charge.
Wait, they just flip their charge?
Exactly. When the scientists are mixing the vaccine in an acidic environment, these lipids take on a positive charge. And since mRNA is naturally negatively charged, the two attract.
Like magnets?
Right. The lipids physically condense around the mRNA, folding it up tightly and protecting it inside this secure shell.
Wow. And what about the other two? The helper lipids and the pegylated lipids?
The helper lipids provide structural integrity. Think of them like the mortar holding the lipid bricks together, ensuring the bubble doesn't just collapse on itself.
Makes sense. And the pegylated ones?
So PEG stands for polyethylene glycol. These act as a stealth coating on the outside of the bubble. This coating keeps the nanoparticles from clumping together in the vial and helps them evade early detection by white blood cells once injected.
So this highly engineered stealth bubble drifts through the body, bumps into a cell, and the cell essentially swallows it up into an internal compartment called an endosome.
That's right.
But it's still trapped in the bubble, right? How does the mRNA actually break out into the workshop?
This is where that ionizable lipid does its second magic trick.
Oh, I like magic tricks.
The cell naturally pumps acid into the endosome to digest whatever it just swallowed. As the environment gets highly acidic, those ionizable lipids shift their charge again.
So they flip back.
Yes. This rapid change in charge disrupts the endosome's membrane. It basically causes the bubble to burst open.
That is wild.
The fragile mRNA escapes the acidic trap and drops directly into the cytosol, the cell's protein-building workshop, completely intact.
Okay, that mechanism is incredible.
Yeah.
But it also raises a massive contradiction for me, and I think it's something a lot of people try to wrap their heads around.
Sure, what is it?
If the entire goal of this technology is shipping a genetic blueprint to our cells, why are we using mRNA at all? Why not use DNA?
The classic DNA versus mRNA debate.
Right, because DNA is the original permanent blueprint molecule. It's vastly more stable. You don't need to keep it in those ultra-cold deep freezers, and it is much easier to ship globally. Why go through all this trouble to build fragile mRNA delivery systems?
This raises a very important question, and the answer gets right to the heart of how human cells are architected. It's all about crossing the nuclear boundary.
The nuclear boundary.
Yeah. You see, the cytosol, that workshop where the proteins are actually constructed, makes up the outer volume of the cell. But our human DNA doesn't live there. Our DNA is locked away inside a highly secure, heavily guarded inner vault called the nucleus.
The nucleus is essentially the master server room of the cell.
Precisely. It's restricted access. If you attempt to use a DNA vaccine, that DNA payload has to cross two incredibly difficult physical barriers.
Okay, the first is the cell wall, right?
The outer cell membrane, yes, just like mRNA, but then it has to navigate through the cytosol and cross the nuclear membrane to get inside the nucleus, because that's the only place DNA can be transcribed into RNA.
That sounds like a lot of extra work.
It is, and that double journey is notoriously inefficient. But more importantly, it introduces a theoretical biological risk. Anytime you introduce foreign DNA into the nucleus, there is a minute but theoretical chance it could integrate into your own human genome.
Oh, and obviously nobody wants to risk accidentally editing the master server.
Exactly, which is why mRNA's greatest vulnerability, its fragility, is actually its most vital safety feature.
Wow, it's a feature, not a bug.
It lives entirely in the cytosol. It never crosses the nuclear membrane. It never goes near your master code. It functions as completely read-only software.
Read-only. I like that.
The cell reads the mRNA, prints the required protein, and then the mRNA naturally degrades and just disappears within a few days. It drives fast, robust immune expression with zero risk of genomic integration.
So we have this brilliant modular chassis. The LNP is the sophisticated delivery truck, and the mRNA is the secure read-only payload.
You've got it.
But now that this basic chassis is proven, where is the field going next? Because reading through this document, I was really surprised to see there is a massive scientific debate happening within the mRNA engineering world itself.
Oh yeah, it's a very active battleground right now.
There are two competing philosophies fighting over the future of this technology.
The debate essentially boils down to two approaches: the pulse versus the amplifier.
Okay, break that down for us.
Well, one camp argues we should keep the design as clean, minimal, and unobtrusive as possible. They focus on designing shorter, highly optimized mRNA strands that act like a precise pulse.
A pulse, just in and out.
Right. The strand goes in, triggers a very specific, brief burst of protein production, and quickly vanishes. This ensures minimal stimulation of the innate immune system.
So keep it simple, minimize the side effects.
Yeah.
But what is the amplifier camp doing differently?
The amplifier camp is looking at the physical limits of that clean approach and asking, how do we generate a massive amount of protein from a microscopically small dose of mRNA?
Oh, they want more bang for their buck.
Exactly. Their solution is self-amplifying RNA, or SARNA.
The text notes SARNA essentially borrows a biological mechanism from alpha viruses, specifically a replicase engine. How does that work without triggering the exact immune tripwires we just talked about?
Well, it's a calculated trade-off. With SARNA, instead of just sending the instructions for the final protein, you also include the code for a molecular photocopier, the replicase enzyme.
Okay, so a blueprint for the couch and a blueprint for a photocopier.
Right. Once this larger strand of SARNA enters the cytosol, the cell first translates the photocopier. This replicase enzyme then grabs the original mRNA strand and starts churning out thousands of copies of it directly inside the cell.
Meaning you can inject a drastically smaller physical dose into the patient's arm.
Huge advantage.
I mean, the source mentions using single-digit micrograms, which is a tiny fraction of a standard dose.
But as you pointed out, there is a catch. Because this RNA is actively replicating and creating double-stranded structures inside the cell, it behaves a lot like an actual viral infection.
Which the body notices.
It does. This triggers a much stronger immune reaction. You get vastly more protein expression from a tiny dose, but the starting molecule is bulkier to manufacture, and the innate immune response can be much harsher on the patient.
And just to complicate things, there is a third contender mentioned in the source, circular RNA.
Oh, circular RNA is fascinating. It's still earlier in the clinical trial phase, but it attempts to solve the stability issue mechanically.
Mechanically? How so?
Normally, mRNA is a linear strand, like a long piece of yarn. The cell's natural disposal enzymes start degrading that yarn by chewing away at the exposed ends.
Okay, that makes sense.
Circular RNA literally chemically binds the two ends of the strand together to form a continuous closed loop.
Like tying the yarn into a circle.
Exactly. Because there are no exposed ends for the enzymes to grab onto, the circular code evades degradation for much longer. It sustains protein production over extended periods without needing the photocopier machinery.
Here's where it gets really interesting, though. If you step back from the microscopic biology and look at the industrial macroeconomics, this debate between the pulse and the amplifier isn't just about cellular mechanics.
No, it's about much more than that.
It is about global manufacturing bottlenecks.
Yeah.
I mean, if SARNA requires a tenfold lower dose to achieve the necessary immune response, theoretically, a manufacturing plant's capacity instantly increases tenfold without building a single new machine.
That is the ultimate industrial prize. If you can drop the dose requirement by an order of magnitude, you radically increase the number of global patients you can treat with the exact same factory output.
But let's talk about that factory floor for a minute. If this is essentially just code, how do we actually print billions of physical copies?
It's a completely new process.
Right. With traditional vaccines, you needed farms of millions of chicken eggs, or these cavernous stainless steel mammalian bioreactors where you were literally keeping vats of cells alive to grow proteins. Where is the fermentation happening for mRNA?
That represents one of the most profound industrial shifts outlined in the document. We have completely relocated the fermentation step. We are no longer fermenting vast volumes of the final product inside living cells.
So if we aren't using living cells as the factory, how is the RNA actually manufactured?
Well, we still use microbes initially to grow the DNA templates and the specific enzymes we need, like T7 RNA polymerase.
Okay, so some biology is still involved.
Initially, yes. But the actual manufacturing of the final mRNA product happens in what's called a cell-free in vitro transcription reactor, or IVT.
Cell-free.
Right. You take the DNA template, the polymerase enzyme, and the raw chemical letters, the nucleotides, and you mix them together in a highly controlled, sterile reactor. The enzyme reads the DNA template and rapidly prints out the mRNA strands entirely outside of any living biology.
Hold on, you lost me a bit there. Let me challenge that premise.
Sure, go ahead.
If the process is cell-free, and we're essentially just printing chemical code in a sterile reactor, why did the world experience such massive supply chain bottlenecks during the global rollout? It sounds like it should be as easy as hitting print on a commercial press.
Because scaling up this process is absolutely not trivial. By moving away from living cells, engineers basically traded complex biological problems for brutal physics and chemistry problems.
Physics problems.
Yeah. In a traditional bioreactor, your main concern is keeping cells alive. You're worrying about oxygen transfer, shear stress, and nutrient feeding. But in an IV reactor, and specifically during the LNP assembly phase, you are dealing with mass transfer, thermodynamics, and extreme fluid dynamics.
Let's visualize that.
Yeah.
How do physics problems actually manifest on the factory floor when wrapping the RNA?
Okay, so the encapsulation of the fragile RNA into those microscopic fat bubbles happens in microfluidic mixing systems.
Microfluidic. Sounds tiny.
Very tiny. Imagine two microscopic fire hoses. One carries the RNA dissolved in an aqueous solution. The other carries the lipids dissolved in ethanol. These streams are fired at each other and collide in a channel narrower than a human hair. The kinetic energy of that collision has to be mathematically perfect.
Narrower than a human hair. Wow.
Yeah. If the flow rate is off by a fraction of a millisecond, or the temperature fluctuates slightly, the resulting nanoparticles come out the wrong size, or they just fail to fold the RNA inside.
So it's incredibly delicate.
Very. Maintaining batch-to-batch consistency relies on controlling the volatile physics of that continuous collision perfectly.
So the bottleneck isn't waiting for biology to grow. It's the sheer physics of flawlessly assembling billions of microscopic delivery vehicles.
Exactly.
And because of these massive physics challenges, the source mentions the industry had to develop an entirely new key performance indicator, or KPI. It's no longer just about sheer volume of product.
No, because volume alone doesn't equal success here. You can't just measure a factory's output by how many kilograms of raw RNA it prints per day.
Why not? More RNA sounds better.
Because of the trade-offs we discussed earlier, where highly productive RNA can trigger severe immune side effects known as reactogenicity. The true manufacturing metric is now effective antigenic productivity per tolerable dose.
Effective antigenic productivity per tolerable dose. That's a mouthful.
It is, but it's crucial. It doesn't matter if your factory can print a metric ton of RNA if the resulting sequence is too reactogenic for a human patient to safely tolerate.
Right. It's a razor-thin balancing act. You have to thread the needle between getting enough protein expression to protect the patient, but not so much that you trigger systemic inflammation.
Exactly.
But here's the kicker. Once you nail that balancing act and you build a functional scaled mRNA facility, the implications are staggering.
They truly are. It changes the economic foundation of biotechnology.
The source points out something incredible. Once a facility is built and validated, the cell line you need to manufacture a completely new drug is just a digital sequence.
That's the real power of the platform.
Right. If a new viral threat emerges, or a doctor wants to target a newly discovered cancer mutation, you don't have to build a new factory or spend 6 months cultivating a new biological cell line. You literally just type a new sequence into the IVT reactor's computers.
You can pivot a massive industrial factory to produce a completely different vaccine in a matter of weeks.
That total decoupling of the physical facility from the specific biological product is unprecedented.
It's never been done before in medicine.
And this is exactly why this matters for you, the listener, because this technology is aggressively moving far beyond infectious diseases. The literature is pointing toward individualized cancer vaccines, treatments for autoimmune disorders, and rare metabolic diseases.
The scope is massive.
It is. And for the investors and startups operating in this space, the document explains that the defensible intellectual property, the real competitive moat, isn't just the genetic sequence of the disease you're targeting.
The sequence itself is almost trivial to replicate at this point.
Exactly. The real moat is built on stackable layers of IP. It's the proprietary chemistry of those ionizable LNPs. It's the highly scalable purification methods.
Oh, the purification's huge.
Right. For example, the text notes that IVT printing is messy. Sometimes the polymerase enzyme stutters and accidentally prints double-stranded RNA.
Which is bad news.
Very bad, because the body's immune system sees double-stranded RNA as a massive viral threat. So the proprietary methods to purify the batch and remove those rogue double strands are a massive IP advantage.
You are basically building a technology stack, just like a software company protecting its proprietary codebase.
If we connect this to the bigger picture, you can immediately see why this is causing a massive geopolitical shift.
Oh, entirely. The source highlights that the United States, Europe, and China are all aggressively pouring capital into building out domestic mRNA manufacturing hubs.
But they are not doing this just to buy a factory for a specific seasonal vaccine, right?
No, not at all.
What are they actually buying?
They are buying a programmable immunological response infrastructure.
That is a big phrase.
It means they are establishing the sovereign domestic capability to say, regardless of whatever novel biological threat or disease profile emerges next, we possess the digital blueprints and the physical microfluidic infrastructure to print the countermeasure instantly within our own borders.
A programmable immunological response infrastructure. That is a dense, heavy concept. And it brings us directly to the overarching theme of this entire deep dive. What we are witnessing is the standardization of an entire biological operating system.
The old ways are really behind us now?
They are. In the 20th century, the basic building blocks of medicine were live cells, inactivated viruses, and massive steel fermentation vats. Now, the primitives, the fundamental building blocks, are digital sequences, chemical caps, untranslated regions, and precision-engineered nanoparticles. So what does this all mean?
It means the next decade of medical science is going to be defined by bridging the gap between discovery and scale. The industry has to take these fragile, incredibly complex molecular processes and turn them into robust, reliable, and almost boring industrial pipelines.
Boring is good when it comes to manufacturing.
Exactly. We're looking at a near future driven by continuous flow IVT reactors that literally never stop printing, and localized microfactories that can be deployed rapidly anywhere in the world.
I do want to pause here and note for our listeners that we are strictly unpacking the engineering and manufacturing perspectives outlined in this specific source document.
Right, we're just looking at what the authors are presenting here.
According to these authors, the debate over the fundamental efficacy of this mRNA platform is largely settled, and their focus has shifted entirely to how society will manage, regulate, and deploy this immense capability.
Exactly. For them, the technical hurdle has been cleared. The focus now is on how far we can push this programmable modality into every corner of human health, from oncology to gene editing.
Which leaves me with one final thought, something I really want you to mull over after you finish listening today.
It's a great thought experiment.
The authors conclude by suggesting that society must actively decide how to wield this operating system. We spent this entire conversation talking about replacing static glass files with dynamic computer code. So imagine a near future where the very definition of the word drug fundamentally changes.
It's totally possible.
If therapeutics are essentially just digital sequences transmitted to localized microfactories, could your local neighborhood pharmacy one day function like a biological 3D printer?
just printing medicine on demand.
Right. Imagine walking in and securely downloading a highly personalized cancer treatment sequence, one that was designed exclusively for your exact biology just that morning by an algorithm and having it printed on-site while you wait. What happens to the structure of global healthcare and what happens to human longevity when life-saving medicine becomes quite literally as instantly transmittable as an email?
You've been listening to Biomanufacturing and Fermentation Technology with Prasad Urnala. Thank you for joining me. Until next time, keep engineering biology into reliable manufacturing.